looking at organic molecules of organisms, organic molecules always include carbon and hydrogen. Those with only hydrogen and carbon are called hydrocarbons, and they'll be named according to the number of carbon molecules there. So organic molecules, as we will see, can contain other atoms as well, but they will always have carbon and hydrogen. The chemistry of carbon itself accounts for the formation of great variety of organic molecules. It's a really versatile building block. Carbon atoms contain 4 valence electrons. That's electrons in their outermost shell. So there's potentially 4 bonding sites on one carbon atom. A carbon atom may share electrons with another carbon atom or other atoms in order to achieve 8 electrons. This satisfies the octet rule. For example, if we're looking at a carbon atom, this shows 2 here and different ways that they can bond. Carbon is atomic number 6, so if you have a single atom, you have 1, 2, 3, 4, 5, 6 electrons in a neutral atom. So if you have 2 carbon atoms, they can form this single covalent bond. Then other bonding sites off of those as well in order to satisfy that octet rule. Or carbon can form a double bond with another carbon atom, or even a triple bond. So we'll see variations on this as we go along. Whenever we look at organic molecules, we're going to be looking at carbohydrates, lipids, proteins, and nucleic acids. 3 of those have specific building blocks to them, or monomers, that create the polymer. So for carbohydrates, for example starch, monosaccharides are the monomers that join together to build the polymer. For proteins, they are composed of amino acids. So you have amino acid monomers that join together to form protein polymers. Similarly, you have nucleotide monomers that join together in synthesis reactions to form nucleic acid polymers. First, let's look at carbohydrates. In general, carbohydrates provide energy and store energy. They're quick fuel and short-term energy storage in organisms. They also play a structural role in woody plants, bacteria, and insects. On our cellular surfaces, they're involved in cell-to-cell recognition. Carbohydrates always contain carbon, hydrogen, and oxygen. The hydrogen to oxygen ratio is always 2 to 1. Just as in water, we have 2 hydrogen atoms to 1 oxygen atom. And this is because carbohydrates are hydrates of carbon. As we'll see momentarily, there are 3 types we're going to look at: monosaccharides, disaccharides, and polysaccharides. Mono meaning 1, di meaning 2, and poly meaning many. Simple carbohydrates, if we're looking at monosaccharides, they are simple sugars with 3 to 7 carbon atoms. And they're named in terms of how many carbons they have. Pentose refers to a 5-carbon sugar. Hexose refers to a 6-carbon sugar. We're going to be concerned with this little formula here, C6H12O6. This is glucose, and represented here. So we can see in this first structure how all of the carbon atoms are shown. We have an oxygen atom here, and they form this ring structure. And why carbohydrates are called carbohydrates, so we have carbon here, and it's hydrated. So if you think of water, that is H2O, and you break that apart into a single hydrogen and a hydroxide, or OH, you can see that this carbon has become hydrated. Also simple carbohydrates are disaccharides. They contain 2 monosaccharides joined by the dehydration reaction that we saw earlier. Examples of disaccharides are maltose, sucrose, and lactose. So reviewing how that dehydration reaction works, if we have 2 glucose molecules here, and we're keeping an eye on this hydroxide molecule here and this hydrogen here. Whenever this bond is going to be created between the two monosaccharides, we see that a water molecule is removed, or it's dehydrated. And looking at how this is a reversible reaction, since we have these double arrows, if we took this from right to left and added water, then we have a hydrolysis reaction or a degradation reaction, where we're taking this disaccharide, we're adding the components of water and breaking apart this bond. Polysaccharides are many monosaccharide subunits that are joined together, and they can be joined together in different ways, as we'll see momentarily. Examples of polysaccharides include starch, glycogen, and cellulose. These are long polymers, the monomer being a monosaccharide. Starch is a storage form of glucose in plants, and this can contain up to 4,000 glucose subunits. You have fewer side branches than glycogen, as we'll compare them momentarily. Glycogen is the storage form of glucose in animals, so this is short-term energy storage for us. Within our liver, glucose is stored as glycogen, and this helps maintain blood sugar levels in between meals. If we're looking at the structure of starch, and so something like a potato that's starchy, and within these cell walls, and we look at a starch granule, and this is an artist's representation of what starch looks like. So we have these monosaccharide monomers, so joined together in these long chains, each of these hexagons representing a glucose subunit, and you can see how they become branched. This is similar to glycogen, which is also a storage form of glucose, but this is going to be in animals rather than plants. So again, we have these glucose subunits or monomers that have been joined together by dehydration reactions or synthesis reactions. We have more branching here of those glucose subunits. Cellulose is another polysaccharide. Rather than being a storage form of glucose, it's a structural component. Cellulose is found in the cell walls of plants. It accounts for the strong nature of the cell walls, and it has a different chemical linkage than starch or glycogen. And so whenever we eat plants, we can digest the starch but not the cellulose. This prevents us from digesting foods with cellulose. Instead, this is roughage or fiber within our diets. So notice, within cellulose, even though it's still made out of these glucose subunits, that the structure is different, and so we have this linkage that is different. You can compare this, figure 2.18, back to figures 2.17 and figure 2.16. So, as we're comparing those, this is more of a structural form for a polysaccharide than as an energy storage.